language that he is alleged to have used.
Hello, Taylor. Hello, John.
Hello, Margaret.
Oh, it looks like people are making their way in here. That's a good sign. Margaret, I'm just going to let you and Nick kind of coordinate however you want to handle the questions till 3:30. Okay, I'll just be jumping between rooms to check on technical issues.
Okay.
Thank you.
Hey, Margaret, good job.
I'm not sure whose voice that was, but thank you.
It's Scott Pavey.
Oh, thanks. Oh, yes. Now, sorry. Now I'm seeing the SP flashing as you speak.
Margaret, do you have time for a follow-up discussion point on what you said in the other room, or is there an agenda you want to execute?
There is no agenda, and so yes, yeah, if you want to sort of continue on with what we started talking in the other room, that'll, I think, yeah, help stimulate some discussion. So that'd be great.
Sure. Well, I just well, and there's something that came up about in the chat too, but type 1 and type 2 errors are really different from each other in eDNA, in the sense that, you know, cross-application, which we can control for bioinformatically and through our testing in the lab, that can cause a type 1 error if it's not done correctly. But a type 2 error is really totally different, because, you know, we can test for efficiency and everything like that in the lab, but it gets down to like how you're sampling. You know, I mean, one fish in Lake Ontario, you're always going to get a type 2 error on that unless you're next to the fish. But then, you know, a river, it depends on the quantity of fish, but at least there's like a linear movement of the water. And so you've got everything upstream. And anyways, it's type 2 errors are going to be a lot more like on the individual project and its design, rather than what we do validation wise and testing wise in the lab.
Oh, yeah, Taylor, you're asking about the list of 60 native and 20 non-native. Yeah, that's mostly, yeah, so that Daniel's group is largely doing the 60 native, and Bob Hanner's group the invasives. Yeah, we do have a list that, Bob, if I remember correctly, that you identified all of the fish species that are currently alien invasive species in Canada, or a few that are on the watch list, so to speak, and those are the ones on your list. And then I think and I'm not sure. I guess Daniel's probably in the activity 3-4 room. Daniel's group is starting on, I think, fishes of Lake Erie and sort of expanding from there. But we have a list. And then each of our regional groups are starting to work up lists that are of importance to us. And I will mention, seeing John's face, and Deepwater Sculpin is definitely at the top of our list in our lab.
Yeah, Taylor, if you're interested, we can share the list. We've got over 80 species that could be invaders in Canada. Some of them non-natives, but some that are Canadian natives in one part of Canada, but invaders in another part of their range. So it's a bit of a mixed bag.
Yep. Well, this is Taylor. Since it's quiet, I'll ask more questions.
Good.
I don't want to dominate conversation about it. So, Bob, you made a comment, maybe in passing, about there being, if there's more room on the chip, then go ahead and include them. I think I was trying to track what was going on and remember an earlier conversation with Daniel Heath before I think this even went into the submission stage. Is the vision that that these detection assays are also loaded onto a high throughput qPCR chip platform, or did you just mean more generally the suite of assays that you might test a sample for? Can you kind of elaborate there if appropriate, or just cut me off if that's not where you're talking about going?
Yeah, no, I think that was sort of the whole point, is to make targeted assays, but then ultimately port them onto a chip. One of the challenges I see with that, though, is they all have to have very similar annealing temperatures in order to be on a single chip, and that may be a bit of a tall order.
Yeah, and that's all super relevant. And so the open array platform is the one that you're imagining that chip would be on for this as well. I'm sorry if I missed that. I know that was the platform for the gene expression work, but for the detection work as well.
I assume so, but I haven't done any chip work before, so I don't know. Margaret may be better able to answer that question, or we might have to go to Daniel.
Yeah, I've assumed it was the same, but I actually don't know. Yeah, that Daniel would definitely have the answer to that.
Hello, my name is John, and I'm a community sample collector. And I have a simple layman's question about the invasive species. Will you be able to get an idea from the sampling whether there's a little bit, maybe a lot more, or a lot of invasive species in a lake? You know, there are interventions that can be done maybe earlier when there's only a few and not much work, maybe not worth doing it if it's heavily infested. Can we be able to get an idea from that too?
I would say yes, yeah, that in one of two ways, that even just with either eDNA signal strength, that if you're getting lots of amplification that in each of your samples, that would suggest you've got a relatively high abundance. The other thing, particularly in the earlier stages, if say you take 20 samples from throughout the lake and all 20 of them are testing positive for the invasive species, that's a bad sign, versus if it's just one spot near the boat launch or something like that. And so in those cases, even just frequency of detection can be at least a good indicator of how relative abundance. Yeah.
Okay, thank you.
We've got a paper that's in submission right now where we were working with some folks around the forebay of a cooling water intake of a power plant, where they were doing surveys of larval fishes, and they would use two different approaches, trying to net them, as well as they had a pump that would suck water in and run them through a filter. And we did eDNA analysis at those same sites where they were netting and pumping, and we saw actually quite a nice correlation with the pumping method. But interestingly, the netting and the pump method didn't show a great correlation between each other, which isn't surprising. You know, I think a pumping method is a bit more objective. But that said, we did see some diurnal fluctuations. And this was something that was interesting to me because, you know, we don't really know how ephemeral that eDNA signal is, but that eDNA signal seemed to fluctuate up and down at the time of day that correlated with their abundance, which I thought was pretty interesting, that we were getting kind of near contemporaneous signal with the abundance of the organisms in that general vicinity. So, you know, we can never tell you absolute abundance, and Scott mentioned earlier, you know, and Margaret as well, it could be dependent on, you know, distance of where you sample from, and that really speaks to the importance of a robust sampling design. But yeah, it does seem as though we can get relative signal intensity, all things being equal, you know, and there's a lot to standardize there. Volume of water, environmental parameters that like pH and things that could impact DNA survivorship. So, you know, the short answer is yes, we see positive correlations, but I think we have to caveat that with, but there could be a lot of other complicating factors.
Okay.
Yeah, this is Scott. So
that's another.
go ahead.
Sorry, if I could ask just another topic. Could this technique be generalized some point or used to identify plant invasive species too? I mean, it's not your mandate to do that, I know, but you know, the technique we talk about so much, myriophyll and other things that, you know, can impact the lake. And I know that there was a lake in Ontario, I wouldn't, near Peterborough, it would be kind of too simple to say, but they noticed an invasive plant species, and the community got together and they removed it physically and burned it. And over 4 or 5 years, they were able to eliminate or apparently eliminate that plant species because they were very, very active very early in that process. And that might be something that'd be interesting for many, many lakes in the future, to tell them, hey, you've got this, you can still do something about it. And because it's early. Is that something that could happen with this technique in the long run?
Short answer, yes. And I know some people, at least somewhere, are doing exactly that. And that actually has identified, a limitation to what we're doing. I mean, we're being fairly ambitious by including all, 200 plus freshwater fishes of Canada, but we are only focusing on fish, so that the same water samples could be surveyed for other invasive taxa, but we aren't. But you're right, that would be another really useful application.
Margaret, it's Chris. Can I jump in on that briefly?
Absolutely.
So we have developed eDNA markers for about 10 invasive plant species. And even though it's not part of GenFish, because Bob was leading the species ID effort, he has the assays we've developed for all of these, as well as for a few invasive inverts. And I wish we had gotten rid of the water soldier close to Peterborough. That's still very much a work in progress. Okay. We're beating it back. We think we prevented an invasion into Lake Simcoe, and we're still keeping an eye on that using both physical surveys and eDNA.
Okay.
Hey, Chris, do you have one for Eurasian Milfoil?
Yes. So this was published.
We've been developing one, and we found that it cross amplifies with a closely related native. Have you guys tested it against closely related? It's robust against that?
No, it's not. We tried 3 different targets. You use the combination of the three and you will get species specific. Any one of them, no, you don't. Barcoding plants is not nearly as fun as it is for animals. But Scott, they are published, and the combination of them, you can winnow it down to just Eurasian.
Okay, great. I'd love to check that out.
And I don't want to derail the conversation, but anybody who's worked with plant eDNA knows it's also not nearly as simple as working with animals. There's much more seasonal variation in shedding in particular. And except for, unless you hit the time of the year where they're dying back and shedding tissues, it's not nearly as sensitive as eDNA on animals either.
I just wanted to pipe in here as well, if I could. Hello, Margaret, Bob,
Hello.
Don, and others. I'm here at the National Biodiversity Cryobank of Canada, which is the intended archive place. And I figured this was a good opportunity to talk about the importance of archiving, because we can look at fish now, and then we can archive these samples, and then, for example, look at plants later, and maybe the technology will continue to evolve such that it'll become more sensitive to plants in future. So I just wanted to put it to you, Margaret. You know, that's one example, but what is the role and importance in your mind of archiving in terms of the larger GenFish project?
Yeah, I think it's critically important, and you've already identified one of the main values of that is that as technology improves, that all of those samples can then be mined for additional information in the future. Also, you know, we're focusing on fish, but then other, 5 years later, somebody might want to know, was this invasive plant or invertebrate present 5 years ago and it hadn't yet been detected? There's so much information that can be gathered from archive samples, either to prevent having to go back and do all the sampling again, or because, as they often say, you can't go back and sample in the past, and so to look at temporal trends, the value of those specimens is invaluable. So, and I know, that organizing all of that, curating all of that is not trivial, but the value of it is, can't be underestimated or overestimated.
And so, right, with timescales in mind, do you picture repeat sampling happening, like every 5 years, 10 years, etc.?
That's a really good question, because that's not what we're doing. We're trying to develop the tools, but that's exactly the ideal, is to be able to have, particularly for species of concern, or that regular monitoring for invasive species, to have a regular biomonitoring program, and being able to have archived samples that presumably many people could take advantage of, so you don't have everybody trying to reinvent the wheel every year. I'm going to go out and collect water samples for this species. I'm going to collect water samples for that species. Have a much more standardized, sort of coordinated effort for biomonitoring.
Speaking for Ontario, that's likely to get built into our sort of biodiversity monitoring going forward. A number of people on the call will be familiar. There's a program called Broad Scale Monitoring. The goal was to sample 1,600 lakes every 5 years. Use a combination of fixed sampling locations and spatially stratified randomized sites, and then this repeats every 5 years. I'm already getting a lot of questions in terms of, can we build in an eDNA sampling component? Which, well, some people would like to replace the netting with eDNA. I'm pushing back against that. But I do expect for our next cycle, which I think is 2 years away, we'll probably be pushed to include eDNA sampling at that point. Having a robust archive would be wonderful.
Chris, in fact, part of our 500 water body survey will be sampling with broad scale monitoring this summer, at the very least in northwestern Ontario, and potentially also in northeastern Ontario.
Yeah, pretty sure Preston Lennox is in this group as well, and think he'd be very keen on Northeast.
So I'd like to raise a couple of points there. Chris had a note in the chat, if you don't have it open, Roger, where he's asking about archiving existing eDNA filters. This is something that I'm a strong proponent of. If you have a couple of filters from a site and we extract one and archive one, alternatively, you can cut them in half and extract half and archive half. In my experience, DNA aliquots tend not to be terribly stable, and they're certainly not terribly robust to multiple freeze-thaw cycles. So, you know, I think the question remains, I can see your fusion freezers in the background, which are vapor phase liquid nitrogen freezers that reach minus 150 C. So, you know, if anything is going to preserve a DNA extract, it's that infrastructure that Roger has. That said, if we are able to send filters, which is great, back to Margaret's point about multiple uses, how would you envision managing that asset? I mean, you could just pull a filter out and send it to someone else, but then it's gone. So would you potentially have the opportunity to use some sort of a standardized extraction protocol and keep some of the extract and send some of that out, as opposed to letting, you know, essentially if we're only banking filters and the whole filter goes out for extraction, then it's only a one-shot archive.
Yeah, those are really good questions, Bob, and I think we have to figure all of that out. And I think more broadly in the biodiversity collection community, we're thinking about those issues. Now, as you know, with a cryo collection like this, there's an expectation that samples will disappear at some point. And so it's a matter of determining, you know, what criteria we use for loaning those things out and making sure that the projects are credible and important. But yeah, we still have to discuss that and really make sure at the point of collection that there's enough for the first researcher and enough for the archive as well, as kind of our first point. You know, I think the same issue applies whether we extract it and send some out. Like, that DNA is still probably going to disappear at some point, and maybe that's okay. Yeah. Lots of questions. Lots of questions. Yeah.
Hey, just following up on that, sure, multiple freeze-thaw cycles are definitely bad for all DNA, but wouldn't you think that extracted DNA without freeze-thaw cycles would be more stable than a frozen filter, just with all the, I don't know, crap and stuff that's also on that filter? Because then you can just aliquot into many tubes, and then you've got, you know, as long as you plan in advance, you've got aliquots you can give lots of different people, and it's, you know, not a whole filter, and there's only one extraction, so it's comparable and such. Yeah,
it's the challenge with that, having, getting an echo. I think the big problem with that approach is just the front end cost of who's going to bear the cost to do that extraction, aliquot out into all the tubes. Then it takes up exponentially more space in your freezer to go from one tube with a filter to, say, 10 tubes of DNA aliquot, which, you know, if there's the potential to do that at CMN, that's tremendous. I know when we set up this kind of a collection at the AMNH, even as well endowed as the American Museum is, it didn't have the funding to have staff at the cryo collection extracting everything, aliquoting out into multiple aliquots in the hope that someday people would use some of them. To your question about the stability of the DNA as an extract versus on a filter, I think that's a great question. I can't honestly answer it, but what I will say is that DNA extracts stored below the glass transition point of water are not stable over the long term. So -80 is certainly better than -20. -20 is certainly better than a home freezer that goes through freeze-thaw cycles to be frost-free. But yeah, anything where you're kind of warmer than about -130 C, you're allowing ice crystals to continue to form over time. And that's exactly why we see, if you think about plating out bacteria, for example, you can throw a plate of bacteria in a -80, pull it out in a few months, restreak another plate from it, and you'll get some survivorship. Out to about 6 months, it's much more hit and miss, and typically a year of a plate being stuck in a -80, all viability is lost, and you can try and streak that stuff out, and it's just, yeah, it's no longer viable. So it's interesting to see that, you know, there's still quite a bit I think we don't know about DNA stability and how best to archive this stuff.
Yeah, and it may be a combination of, like, so that you have 2 extra filters collected, you know, one for the immediate question, one that gets extracted and aliquots get archived, and then the third filter is stored as a filter, so that you sort of have all bases covered. Some of what we have to do is understand, yeah, the pros and cons of different methods. If one really is, you know, better for stabilizing the DNA, but it may be, you know, there's pros and cons, and, you know, both might ultimately be an approach. Yeah.
It might speak to the advantage of a regional approach to archiving, just because if you had one central in all of Canada, then it would be kind of, it would get monstrous, and it would be hard to prioritize, and, you know, that's just a thought there.
Yeah, I can speak to that. I see costs and benefits of that for sure. I mean, it's definitely good to have more people doing this, since here at the CMN, you know, we do have finite resources. That said, we do have the mandate to keep things in perpetuity forever, which is pretty unique to museums having that mandate. And so if there were a regional approach, I would at least advocate for us getting like a subsample of some of those regional things to archive again in perpetuity.
I would absolutely agree with that, and just, you know, instruct people to look over Roger's shoulder back at that liquid nitrogen vapor freezer. It gets us the best of all worlds by being well below that glass transition point of water. And the other thing that I love about that technology is the vapor nitrogen in there. In the face of a power outage, you've still got a week or more of liquid nitrogen in that freezer. So in terms of fail-safe, it's far better than the crappy -80s that always seem to blow out whenever we're on vacation or holiday in our labs. So I absolutely agree with Roger. And, you know, and the sad reality is we don't have a budget for doing this kind of archival within the Ginfish project per se. So my perspective is we're damn lucky to have the CMN with some funding and a mandate to do this.
Well, you know, I think the issue is no one has the budget to do it. And it hasn't just been, it's not just recent genetic samples. Just look at museums in general in Canada with conventional vouchers, right? They've been diminishing over time as a result of inadequate resources. The question is, how do we turn that around? Maybe it's when they see more than just, this is more than stamp collecting for species, but rather there's the applications that we're developing in the other, you know, the other activities as well, that they might start realizing, wait a minute, this is, yeah, you know, this is just not simple stamp collecting. It is measuring items of you know, natural and economic importance over time.
Yeah, you're preaching to the choir on that one, and I would say, and Roger knows this well, you know, trying to advocate for funding for natural history collections is a problem in the best of times. I'm not aware of any kind of coherent lobbying in Canada to do that. A number of my colleagues at the Smithsonian have put out several different reports there to Congress trying to see the kinds of budget lines for natural history collections that have been set aside for, say, GenBank from the National Library of Medicine. You know, it's that, I think we would all agree, it's that same kind of infrastructure, you know, and so if we want to think about maybe as part of our GELS activity that, you know, a group of the PIs, I mean, shoot, we've got, you know, nearly 100 people just at our AGM today. an open letter to INSERC and essentially anyone who will listen, I think is a worthwhile endeavor.
We have to remember that thought. That's an excellent thought.
Yeah.
Yeah.
They would, besides the agencies, I'd also say sending to some of the federal ministers, that as well as chief scientists.
Yeah. I'll just mention, I think our strength here at the Museum of Nature, our main strength is the mandate that we have and the history that we have and the credibility. But of course we are underfunded, especially in the context of COVID, where we're not getting any admissions through the door. But we are working with partners, specifically on the eDNA things, but to be a genomic repository for places like Parks Canada. And we look at those people as having much more, deep pockets than we do, but without the mandate that we have. So we really want to partner with folks like that. And there's other people in the conversation here who represent organizations that might have deeper pockets than we do as well. So I think there's lots of opportunity. We've also launched just within the past few months a new foundation, the Nature Foundation, specifically to raise funds for the Museum of Nature. And so we're always looking for kind of sellable sponsorship opportunities. And I think there's opportunities in this for that as well.
The other way to think about it is that everyone just adds vouchering costs to their the front end, to their sampling. When I was at DFO, I always paid the ROM to deal with our conventional vouchers, because DFO should not be not managing their own collection. They should leave it to the professionals to do, right? And I think just because few people are trained within the museum environment these days, they don't think about the vouchering aspect, right? It's an afterthought rather than to me, it's front of mind. And that's one way to actually fund some of this. But then museums have to come up with and other facilities that do this have to come up with, economic models to make this viable, where you say we're going to add 10% or whatever, and then you're going to look after the sample in perpetuity as a result, right?
And that gets us into a pretty sticky wicket. I remember when my time from the American Museum, we thought about putting a small handling fee on just pulling a sample, the time it takes for a tech to pull a sample out of the freezer, subsample it, package it up, ship it off. And the museum community in the US lost its mind because, and perhaps it was a bit of an illusion, but people were saying, well, you're profiting off of genetic resources. And, we're saying, no, there's no profit margin here. This is purely cost recovery. But, again, that's sort of the double-edged sword of going back to the gels people, is they can spin it out in directions you didn't anticipate.
Oh, I think, Bob, like you've really hit the nail on the head here with the gels group. This whole area is one that I don't think we really thought that much of before this discussion. But I think it's a critical area where that we need to address, or else a lot of this will be for naught, right? It'll be for individual projects, but not for collective future and natural resource management in Canada.
Bob, just lump it in with the postage. Don't break it out as a handling fee.
Shipping
guys,
and handling fee.
do you mind if I jump in for a second here?
No, go ahead, Joe.
The discussions are going very well. The informal session is scheduled to start at 20 to 4, but we only have like 40 some odd students who will present. Do you guys want to keep wrapping for another 15 minutes or so? And then we can start the informal session at 4? Sounds like you're making some good progress. It's up to you guys.
I think we're adaptable, that if other groups are ready to switch to the informal, because
there's a 20-minute break built in.
Yeah.
We do have a 20-minute break built in at this time. So
okay, yeah.
Joe, why don't we just go ahead and stick to the original schedule, just for the Zoom fatigue element on the tail end of this.
Okay. So what I'd like you guys to do then is, on this screen, if you click the leave button, You should then see a window with the original formal room. You will have to click resume to rejoin the room.
Okay.
Okay, thank you very much, and I will stop recording now. Thank you.
I'm going to have to go and pick up kids, so I'm not going to